Hello students, today we are going to deal the first part of Unit 4 summary, a deal about food making and growth in plant. We mainly focused on the leaf and photosynthesis. Let's start. As you know, food making and growth in plant. Plants have different organs. Especially, let's see, flowering plant have different organs, and non-flowering plant also. For the main organ of the plant are flowers, a reproductive organ, and leaves, stems, and root are the main organ of the plant. When you say flower, for flowering plant, and cones for non-flowering or gymnosperms. So let's start about the leaf, a photosynthetic machine. In the leaf, what makes the leaf? It's a photosynthetic machine. What makes the leaf adapt to make food? What does the leaf to make food? These are the most important adaptation of the leaf. The first one is large surface area to absorb maximum amount of light, because light is very important in photosynthesis. The second adaptation of is the leaf are thin, hence it is a short distance for gas exchange. You know, in photosynthesis, plant take carbon dioxide from the atmosphere and release oxygen. So it is very important, its thin size is very important adaptation. The other, it have a network of veins to transport substance, to transport water, to transport gases, and to transport water from the root. So it's the most important adaptation. And the presence of opening stomata. The leaf have an opening of stomata used for exchanging of gases. The most important adaptation of the leaf for photosynthesis. The most important adaptation of the leaf is the presence of large amount of chlorophyll. The presence of large amount of chlorophyll is very important adaptation of the leaf to make food, because chlorophyll is very important to absorb solar rays, light energy, which is very important to split water molecule into hydrogen and oxygen. So these are the most important adaptation of the leaf to make food for photosynthesis. So let's see the external and internal structure of the leaf. Let's start from external structure of the leaf. As you see here, the leaf have epithelial or leaf stalk. The one which attaches the leaf to the stem, that part is called epithelial or leaf stalk. Epithelial or leaf stalk, which is used to attach the leaf to the stem. The other one is a lamina or leaf blade. This one, the most photosynthetic area of the leaf. The other one, midrib and the veins. You see this one, a midrib and the vein, which is very important to transport substance. And there is margin. You know, the leaf can divide based on the margin. So the leaf have its own external structure. That is very important adaptation to make the leaf food, to make food for photosynthesis. The other important is internal structure of the leaf. Let's start the internal structure of the leaf. The leaf have, when we go to the internal, it has this kind of internal structure. Let's start from the upper to the lower. From the upper of the leaf, as you know, the upper part of the leaf is very green because of the presence of waxy cuticle. And then there is epidermis. So this is the internal structure of the leaf. In internal structure of the leaf, on the upper part there is a cuticle, and then epidermis, and there is a regularly arranged cell, a palisade mesophile, and a loosely arranged cell, a spongy mesophile, and the lower epidermis, and the guard cell. In the lower epidermis there is a guard cell and stomata, an opening. So these are the internal structure of the leaf. Now we are going to deal each parts. Let's start from a waxy cuticle. The outer thick waxy covering of the leaf found on the surface of the leaf is called, cuticle. which is very important to prevent water loss from the leaf by using transpiration. On the upper part of the leaf, there is a cuticle which is very important to prevent water loss from the leaf. And the second part, internal structure, the mesophile. There are two types of mesophile. Next to cuticle, there is epidermis, and this one is a mesophile. Mesophile, palisade mesophile, and spongy mesophile. Let's start the palisade mesophile. You see, the upper one is a palisade, and this one is spongy mesophile. What make palisade mesophile and spongy mesophile differ? There are two types of mesophile, the palisade mesophile and the spongy mesophile. Let's see. On the palisade mesophile, they have closely packed cell. This one, the palisade mesophile. It is closely packed cell, whereas the spongy mesophile is a loosely packed cell. What other difference? They have many chloroplasts. Look at, on the palisade mesophile, the green one is a chloroplast. The yellow is a nucleus. They have many chloroplasts. What is our conclusion for this? The presence of many chloroplasts make the photosynthesis, the presence of many chloroplasts. Palisade mesophile is the main area of photosynthesis. But in the palisade spongy mesophile, there is a chloroplast. It can make photosynthesis, but it has few chloroplasts. What other difference? On the palisade mesophile, as you see, there is no air space. Little air space. It has less air space. But in the spongy mesophile, it has a lot of air space. So, the presence of air space is very important for gas exchange. What do you mean gas exchange? It takes carbon dioxide to make food, and it releases oxygen using respiration. It releases oxygen, photosynthesis. So it's very important to exchange of gases because of the presence of air space on the spongy mesophile. The other important thing, when you see the shape, the palisade mesophile is a regularly arranged cell, whereas the spongy mesophile is irregularly arranged. So, the palisade mesophile and the spongy mesophile is the most important internal structure of the leaf, which can make food. Mainly, the palisade mesophile is the main area of photosynthesis, whereas spongy mesophile can make food, photosynthesis, but it has air space in addition to making photosynthesis. The presence of air space in the spongy mesophile is very important for gas exchange. So the other part is in the lower epidermis. We have learned the cuticle, upper epidermis, palisade mesophile, spongy mesophile, on the lower epidermis. In the lower epidermis, it has a single layer and contain an opening known as stomata. And stomata have guard cell. You know, the opening, it's opening, and the opening contain, these two are a guard cell, and there's opening, it's called, the opening is a stomata. So stomata is very important, used for gas exchange. So the guard cell controls the opening and closing of the stomata. The question is, think about how guard cell control the opening and closing of the stomata. Very nice. Guard cell controls the opening and closing of the stomata. During the daytime, photosynthesis is very high. So what? Concentration of sugar in the guard cell is very high. During the daytime, due to the because of the presence of much light, there is high photosynthesis. High photosynthesis means much sugar is produced because of osmotic effect. Water get into the guard cell, and the stomata swells, and the guard cell swells, and the stomata open. So, during the daytime, because of high photosynthesis, there is high sugar is synthesized. As a result of osmotic effect, water enter to the guard cell, the stomata swells, the guard cell swells, and the stomata open, just like this. During the daytime, during the night time, because of less light, photosynthesis is low. Sugar is low in the guard cell. As a result of this, water moves from the guard cell. The stomata shrink, the guard cell shrink, and the stomata close. So stomata, guard cell controls the opening and closing of the stomata because of the presence of the synthesis of sugar in the guard cell. During the daytime, high sugar is synthesized and water enters to the guard cell, stomata open. During the night time, less sugar is produced because of less light. As a result of that, water lost from the guard cell and stomata, the guard cell shrink and stomata close. So this is the opening and closing of the stomata. So the other one is a vascular bundle, the internal structure of the leaf. We have learned the cuticle, the palisade mesophile, the stomata, and now we are going to deal about the vascular bundle. Vascular transport system. It contains xylem and phloem. Xylem transport water. Phloem transport phloem, important in transport synthesized food to all parts of the leaf. So xylem transport water and ion from the root to the stem to the leaf, whereas phloem transport synthesized food all from the leaf to all parts of the leaf. These are the vascular bundles. So let us go into deal about photosynthesis. We have learned about the leaf, internal structure and external structure of the leaf, and the adaptation of the leaf for photosynthesis, because photosynthesis takes place inside the leaf. And now we are going to deal about photosynthesis. What do we mean photosynthesis? Photosynthesis is photo means light, synthesis means making. So when we combine the two, photosynthesis is a process of making food by green plants. Photosynthesis means a process of making food by green plants in the presence of light and chlorophyll. Let's see, you have seen this chemical equation. Carbon dioxide plus water in the presence of light and chlorophyll produce glucose and oxygen. Oxygen gas is liberated in the process of photosynthesis. So what do we mean? What do we conclude from this equation? We conclude that in photosynthesis, there is two raw materials, carbon dioxide and water. In photosynthesis, two products, glucose and oxygen. In photosynthesis, two conditions, light and chlorophyll. So in order to make photosynthesis, in order to make glucose and oxygen in photosynthesis, four things are required. Carbon dioxide, water, light, and chlorophyll are required in order to make food by photosynthesis. So in photosynthesis, we have said, plant draw up water from the soil and taking carbon dioxide from the air. It draw up water from the soil and take carbon dioxide from the air, and the leaf trap energy from the sunlight and make sugar and release oxygen. So plant use sugar for growth and asparagus. So we conclude that plant, we have said it's a four requirement. It draw up water from the soil, it take carbon dioxide from the air, it use sunlight as a source of energy, and make sugar and release carbon dioxide as a byproduct. That is. So we conclude that the primary product in photosynthesis is glucose, but plant use its primary product for other purpose, for other organic molecule made inside the plant are made as a result of its from primary product glucose. You know, glucose is a primary source of energy. Glucose used for growth. Glucose used for repair of tissue. And glucose converted into starch for storage. The question is, plant make its primary product in the form of glucose, but do not store its primary product in the form of plant, do not store its carbohydrate in the form of glucose. It is stored in the form of starch. Why? Because glucose have osmotic effect. Because of its osmotic effect, since it is soluble. Starch is insoluble, do not have osmotic effect. Plant store its carbohydrate in the form of starch. So the question is, how do we test the plant contain starch or not? We can test using this experiment. First, cut the leaf, and then boil the leaf in hot water, and then boil the leaf in ethanol. Why we put the leaf in ethanol? What is the purpose of boiling the leaf in water? And then rinse in hot water and test the presence of starch in using iodine solution. If it give a blue-black color, the plant contains starch. If it did not give a blue-black color, the plant did not contain starch. The question is, why we boil the plant in leaf in hot water? Why we boil the leaf in hot water? What is the purpose of boiling the leaf in ethanol? Think about it. And then, because the leaf is put in the boiled water because to stop all the biochemical process and by killing the leaf and make the leaf sought for iodine penetration. Why we put the leaf in ethanol? To remove a green pigment. And the positive result is a blue-black color, is a positive result in iodine solution. So let's do this one. I would give 1 minute, 30 seconds. In testing the leaf for starch, the leaf put in ethanol because, very nice. If you say to remove a green pigment from the leaf, you are right. So, phase of photosynthesis. Photosynthesis have two phases, the light-dependent and the light-independent phase. The first phase of photosynthesis is light-dependent phase. What do you mean light-dependent phase? As the name indicates, it depends on light. Chlorophyll absorbs a radiant energy, and the chlorophyll converts radiant energy into useful form of chemical energy. In light-dependent reaction, there is water molecule split, photolysis of water into hydrogen and oxygen, and generate high amount of heat energy. In the light-dependent reaction, water molecule is split, radiant energy converted into chemical energy, and hydrogen ion is produced. This is very important to take place light-independent reaction. Photosynthesis have two phases, light-dependent and light-independent. The first one is light-dependent. As the name indicates, it depends on light. A chlorophyll absorbs radiant energy and converts radiant energy into useful form of chemical energy. In light-dependent reactions, the most important thing, a split of water molecule into hydrogen and oxygen. In the light-dependent reaction, it generates ATP, used as a source of energy. And this is a chemical equation, water molecule is split into hydrogen and oxygen. So in light-dependent reaction, it generates ATP, and it produces hydrogen ion. This is very important to continue the second phase of photosynthesis. In the light-independent phase, it does not require light. It takes place during the daytime, during the nighttime. The only thing, it does not require light. It depends on hydrogen ion, which is produced in light-dependent reaction in carbon dioxide. And carbon dioxide is reduced into sugar. Used ATP as a source of energy, which is produced in the light-dependent reaction. So, in order to take light-independent reaction, it needs hydrogen ion from light-dependent reaction and ATP from light-dependent reaction, and make glucose as it produces glucose, organic molecule is produced. So, what are the most important requirements in photosynthesis? We will see. The first one is carbon dioxide. Carbon dioxide is a source of carbon to prepare sugar, and plant get carbon dioxide from the air. Plant taking carbon dioxide from the air. And how do we know carbon dioxide is required for photosynthesis or not? using this experiment. As you see in this experiment, potassium hydroxide, or sometimes we can use sodium hydroxide, soda lime, it absorbs carbon dioxide. In this flask, use potassium hydroxide. On the second flask, use this sodium hydroxide or potassium hydroxide used in the first flask. On the second flask, on the first flask, we use sodium bicarbonate. You know, sodium bicarbonate generate carbon dioxide. Potassium hydroxide, it take carbon dioxide from the atmosphere. Finally, we test the presence of starch in these two flasks. The one which generate carbon dioxide, this flask shows a blue-black color, positive for starch. And the second one, so the presence of potassium hydroxide, since carbon dioxide is absorbed, it do not give positive result for starch. So carbon dioxide is very important in photosynthesis. The second requirement, the chemical substance used to absorb carbon dioxide in the activity to show the importance of carbon dioxide in photosynthesis is A, iodine solution, B, potassium hydroxide solution, C, Benedict solution, D, copper sulfate solution. I hope if you say potassium hydroxide solution, you are right. The second requirement in photosynthesis is water. Water is a source of hydrogen to make organic molecule. So the other important thing, the oxygen gas liberated in photosynthesis, where does it come from? It comes from carbon dioxide? It comes from water? So oxygen gas liberated in photosynthesis comes from water. And how do we know water is required in photosynthesis or not? By using heavy water, the one which contains a radioactive isotope oxygen. Look at this experiment. As you see in this experiment, in the first experiment 1, carbon dioxide plus heavy water, which contain oxygen isotopes, it liberates oxygen isotope. So we conclude from this experiment, water is required for photosynthesis. And we can conclude also, the oxygen gas liberated in photosynthesis comes from the water molecule. And also we can use the similar oxygen isotope for carbon dioxide. As you see, the oxygen here, glucose, produce oxygen isotope. So the carbon dioxide, the oxygen did not liberate from carbon dioxide. The oxygen liberate from the water molecule. This is very important. We use a heavy water in order to we can test the presence, the requirement of photosynthesis, water for photosynthesis. The other third requirement is sunlight. You know, sunlight, light energy is very important to break down water molecule into hydrogen and oxygen. How do we test whether is required, sunlight is required for photosynthesis or not? By using discharging the plant, covering the plant in black paper and leave for 24 hours and cut the leaf, test the presence of starch. When we test the presence of starch, the one which is discharged or prevent to obtain light give a negative result. That give a negative result. The other which exposed to the light give a positive result, blue-black color. So we conclude that light is important in photosynthesis. So we have seen in photosynthesis, we need carbon dioxide is required, water is required, and now we have seen sunlight is required. What about? And the other important thing is, as a result of light energy, water molecule is split, photolysis of water, and release oxygen. How do we know oxygen is liberated or released in photosynthesis? Using this experiment. In this experiment, there is a pond width like hydrilla, and put in the flask a lot of water, put water, and the inverted funnel. We used this water plant because it generates a bubble of oxygen gas. After a few hours, there is oxygen is accumulated in the funnel. Then if we test this, there is a glowing split is obtained. This is, we conclude that oxygen is released in photosynthesis. The fourth requirement in photosynthesis, we have seen water, carbon dioxide, and sunlight is required in photosynthesis. So how do we know oxygen is liberated in photosynthesis? By using this experiment. There is a pond with in the water. The pond with generate high amount of a bubble of oxygen gas, and oxygen gas is accumulated inside the flask. When we tested the presence of oxygen, there is a relight of a glowing split is happened. This we can conclude oxygen is liberated in photosynthesis. Oxygen is released in photosynthesis. The other requirement in photosynthesis is chlorophyll. We have seen carbon dioxide, water, and sunlight is required in photosynthesis. The fourth requirement is chlorophyll. You know, chlorophyll is a green pigment which is found in the chloroplast of a cell. Chlorophyll used to trap light energy. It have a green pigment which used to trap light energy. So how do we know chlorophyll is important in photosynthesis or not? Using variegated leaf. Variegated leaf means the leaf that have different colors. As you see here in this leaf, it have green color, yellow color. I hope you have experienced this kind of leaf. The green color shows that the presence of chlorophyll. The yellow color shows that the absence of chlorophyll. When we test this leaf for the presence of starch, only the green color shows a blue-black color. The yellow color do not show a blue-black color. So we conclude that only in the green part make food, the yellow part do not make. Chlorophyll is important in making photosynthesis. So the importance of photosynthesis. Even though photosynthesis have a number of importance, we can divide photosynthesis importance into two. The first one is photosynthesis is a source of energy for all organisms. Whether the source of energy for directly or indirectly. It also a source of fresh air. As a result of photosynthesis take high amount of carbon dioxide from the atmosphere. So what? As a result of taking in high amount of carbon dioxide. Carbon dioxide is a greenhouse gas which causes a global warming. So photosynthesis is involved in preventing against global warming. And it also generates high amount of oxygen. Generate high amount of oxygen. So photosynthesis have a source of energy, is used as a source of food, and it gives fresh air. By taking in carbon dioxide, it would cause global warming and release oxygen, which is very important. So these are our today's lesson. Let's sum up what we have learned. Today, we have learned about the leaf and photosynthesis. We have learned the leaf have different adaptation, just like large surface area, its thin, the presence of stomata, the presence of chlorophyll are some of the important adaptation of the leaf. It also, the leaf have its external structure also have one mechanism of adaptation. For the leaf to make food, these are the presence of the leaf exposed to the sunlight because of the petiole and the lamina, the margin is the external structure of the leaf. We also learned the internal structure of the leaf. The leaf have a cuticle on the upper surface of the leaf to prevent water loss from the plant. In the leaf have its internal structure a mesophile, the palisade mesophile and the spongy mesophile. We compare what makes the palisade and the spongy mesophile differ. The leaf have, on the lower side of the leaf, there is opening. That opening is called stomata. The opening is called stomata. And we have also learned how stomata open and close during the daytime, during the night time. We have learned photosynthesis. What is photosynthesis? We have said photosynthesis is the process of making organic molecule from simple inorganic molecule in the presence of carbon dioxide and water. We have learned how the presence of starch in the leaf. How do we test the presence of starch inside the leaf? By using iodine solution. Iodine solution is a reagent which is used to test the presence of starch inside the leaf. We also learned the different phase of photosynthesis, the light-dependent phase, the light-independent phase. What main things happen in the light-dependent phase? Photolysis of water, and it releases ATP, a source of energy. In the light-dependent reaction, reduction of carbon dioxide into sugar. Reduction of carbon dioxide into sugar, a light-dependent reaction. It uses hydrogen ion, which is liberated in light-dependent reaction, and ATP in the light-dependent reaction. So photosynthesis have two phases, the light-dependent and the light-independent phase. What are the most important requirements in photosynthesis? In order for photosynthesis to make food, it needs 4 requirements. Carbon dioxide, water, sunlight, and chlorophyll are needed to make photosynthesis. We also learned what are the importance of its photosynthesis. Even though photosynthesis have a lot of importance, we can divide into 2. It gives us a source of energy. It gives us a fresh air by taking in carbon dioxide and by releasing oxygen. This is what we have learned today. This is the lesson cues. The first one, plant convert its primary product photosynthesis, glucose to starch. Because A, because starch is important in making cell wall of plant. B, because large amount of glucose become toxic for the plant. C, because starch does not affect water balance. D, because glucose cannot be used for the experiment directly. Very nice. If you say because starch does not affect water balance, you are right. As you know, glucose is soluble, so it have osmotic effect. Starch is insoluble, it do not have osmotic effect. So plant stores their primary product, glucose, into starch, convert into starch, and store in the form of carbohydrate, store in the form of starch, not in the form of glucose. So the answer is C, because starch does not affect water balance. Question number 2. In one experiment conducted to prove the necessary condition for photosynthesis, variegated leaf are used. The use of variegated leaf to show the presence of A, chlorophyll, B, carbon dioxide, C, light, D, water. Very nice. If you say chlorophyll, you are right, because variegated leaf are the leaf that have different colors. So the green color shows the presence of chlorophyll, so variegated leaf are used to show the use of chlorophyll for photosynthesis. This is our today's lesson. Wish you a nice day. Goodbye.